Hello students, how are you doing? Welcome to another wonderful biology class. Today we will discuss about part 2 of a cell organelle called mitochondria. Before we start with the lesson, make sure that you have subscribed to this channel and please be kind to like and share this video. Alright, let us begin. Mitochondria structure. Mitochondria may have a number of different shapes. A mitochondrion contains outer and inner membranes composed of phospholipid bilayers and proteins. The two membranes have different properties. Because of this double-membraned organization, there are 5 distinct parts to a mitochondrion: the outer mitochondrial membrane, the intermembrane space, the space between the outer and inner membranes, the inner mitochondrial membrane.
The cristae space, formed by enfoldings of the inner membrane, and the matrix space within the inner membrane, which is a fluid. Mitochondria have folding to increase surface area, which in turn increases ATP, adenosine triphosphate, production. Mitochondria stripped of their outer membrane are called metaplasts. Outer membrane of the outer mitochondrial membrane, which encloses the entire organelle, is 60 to 75 angstroms, with circle thick. It has a protein to phospholipid ratio similar to that of the cell membrane, about 1:1 by weight. It contains large numbers of integral membrane proteins called porins. A major trafficking protein is the pore-forming voltage-dependent ion channel, VDAC. The VDAC is the primary transporter of nucleotides, ions and metabolites between the cytosol and the intermembrane space. It is formed as a beta barrier that expands the outer membrane, similar to that in the gram-negative bacterial membrane. Larger proteins can enter the mitochondrion if a signaling sequence at their N-terminus binds to a large multisubunit protein called translocase in the outer membrane, which then actively moves them across the membrane. Mitochondrial proteins are imported through specialized translocation complexes.
The outer membrane also contains enzymes involved in such diverse activities as the elongation of fatty acids, oxidation of epinephrine, and the degradation of tryptophan. These enzymes include monoamine oxidase, rotenone-insensitive NAD cytochrome C reductase, kynurenine hydroxylase and fatty acid coligase. Disruption of the outer membrane permits proteins in the intermembrane space to leak into the cytosol, leading to cell death. The outer mitochondrial membrane can associate with the endoplasmic reticulum membrane, in a structure called MAM, mitochondria-associated membrane. This is important in the mitochondria calcium signaling and is involved in the transfer of lipids between the and mitochondria. Outside the outer membrane are small, diameter, 60 with circle, particles named subunits of Parson.
Intermembrane space of mitochondrial intermembrane space is the space between the outer membrane and the inner membrane. It is also known as perimitochondrial space. Because the outer membrane is freely permeable to small molecules, the concentrations of small molecules, such as ions and sugars, in the intermembrane space is the same as in the cytosol. However, large proteins must have a specific signaling sequence to be transported across the outer membrane, so the protein composition of this space is different from the protein composition of the cytosol. One protein that is localized to the intermembrane space in this way is cytochrome C. Inner mitochondrial membrane. The inner mitochondrial membrane contains proteins with 3 types of functions, those that perform the electron transport chain redox reactions.
ATP synthesis, which generates ATP in the matrix-specific transport proteins that regulate metabolite passage into and out of the mitochondrial matrix. It contains more than 151 different polypeptides, and has a very high protein to phospholipid ratio, more than 3:1 by weight, which is about 1 protein for 15 phospholipids. The inner membrane is home to around 1/5 of the total protein in a mitochondrion. Additionally, the inner membrane is rich in an unusual phospholipid, cardiolipine. This phospholipid was originally discovered in cow hearts in 1942, and is usually characteristic of mitochondrial and bacterial plasma membranes. Cardiolipine contains 4 fatty acids rather than 2, and may help to make the inner membrane impermeable, and its disruption can lead to multiple clinical disorders including neurological disorders and cancer. Unlike the outer membrane, the inner membrane does not contain porins, and is highly impermeable to all molecules. Almost all ions and molecules require special membrane transporters to enter or exit the matrix. Proteins are ferried into the matrix via the translocase of the inner membrane, TIM, complex or via OXA1L. In addition, there is a membrane potential across the inner membrane, formed by the action of the enzymes of the electron transport chain. Inner membrane fusion is mediated by the inner membrane protein OPA1.
Cristae. The inner mitochondrial membrane is compartmentalized into numerous folds called cristae, which expand the surface area of the inner mitochondrial membrane, enhancing its ability to produce ATP. For typical liver mitochondria, the area of the inner membrane is about 5 times as large as the outer membrane. This ratio is variable in mitochondria from cells that have a greater demand for ATP, such as muscle cells, contain even more cristae. Mitochondria within the same cell can have substantially different crystal density, with the ones that are required to produce more energy having much more crystal membrane surface. These folds are studded with small round bodies known as F1 particles or oxysomes. Matrix, mitochondrial matrix. The matrix is the space enclosed by the inner membrane. It contains about 2/3 of the total proteins in a mitochondrion. The matrix is important in the production of ATP with the aid of the ATP synthesis contained in the inner membrane. The matrix contains a highly concentrated mixture of hundreds of enzymes, special mitochondrial ribosomes, terna, and several copies of the mitochondrial DNA genome. Of the enzymes, the major functions include oxidation of pyruvate and fatty acids, and the citric acid cycle. The DNA molecules are packaged into nucleoids by proteins, one of which is TFAME. That is all for today, folks. One last thing. Before you go, make sure that you have subscribed to our YouTube channel and be kind to like and share this video. This video is edited by Baruch Solomon M.Sc and Genetics. I will see you in the next video. Goodbye!